Okay, so Sihe, you can start saying something.
Okay, so this is the lecture of the series of lecture that Dr. Hartman is going to give here. As we all know here, Dr. Hartman is a more famous scientist coming from MIT to Caltech for visiting for several weeks. And he has very deep knowledge about where we are from and why we are here. So yeah, Dr. Hartman, you can just start.
Pardon?
Yeah, I think it's good time for you to start.
I can start?
Yeah.
Okay. So I'm happy to be in sunny California, as you could see, and that I'm trying, this is an experiment, to deal with molecular biology in a planetary academic configuration. So I want to start off with the problem of the genetic code, its origin and evolution. It's a small topic. Is this moving? Hello?
No, it's frozen.
Hold it. You? Hello?
One second. What?
Okay, now.
Yeah. Okay, sorry.
So in 1966, Francis Crick presented the genetic code. Now, since I'm talking to people that may not know very much about molecular biology, let's talk about the actors in this genetic code. The first set of actors are the amino acids, and they are the stuff that goes to make up proteins. And they come in various varieties. Two of the most important varieties are the hydrophobic amino acids and the hydrophilic amino acids. And then there are the special amino acids. The hydrophobic amino acids have the side chains in green, and those side chains in the hydrophobic cases are, those side chains do not like to be in water. The hydrophilic side chains, on the other hand, like to be in water. And the special ones have things that either have no side chains or a very odd one called the amino side chain. So these are the actors. Now, what do these actors do? Well, they form polymers. And they form polymers. Here's two of these amino acids, and they get rid of water and form the peptide bond. And then you can see various configurations of these peptides. One is a beta hairpin, and the other is oxytocin. That's why you hug your significant other. And then there is the alpha helix. So these are peptides. Now, what do peptides, do we know anything about peptides? Well, yes, we do. There's an enzyme called lactase. Lactase breaks lactose, the sugar in milk, and to glucose and galactose. And this is lactase. And unfortunately, I do not deal, my lactase enzyme doesn't exist. It doesn't work. So I'm lactose intolerant. And here you could see on the upper cart is the human one that's functional, but there's mutants and they are non-functional. You can see a big piece of it is missing, both in A and C. Now, the other things, these are they have rat lactases and chicken galactases. And the other thing you'll notice is that there's a lot of very common amino acids. Now, this is a very big enzyme. It's got 1,603 amino acids, all in one big peptide. So this is an interesting enzyme, and it shows you that these peptides can get very big and be very powerful. Now, the other players in this game are the 4 nucleotides: uridine monophosphate, adenine monophosphate, cytidine monophosphate, and guanidine monophosphates. And they form phosphodiester bonds. And in DNA, the sugar is dioxy as opposed to having 3 and 5 prime. Anyways, 2 prime and 3 prime. But anyways, these can hydrogen bond. A goes to T and G goes to C. And this is what allows you to have DNA or RNA double helix. Now, there are 3 RNAs that I want to talk about today. One is the ribosomal RNA in the large and small subunit. The other one is the tRNA, the transfer RNA, which acts as an adapter between messenger RNA and amino acids, and messenger RNA, which in the case of lactase is a very long piece of RNA. Because you'll find out that the big idea is that happened here in Caltech, which is called the 1 gene, 1 enzyme hypothesis. George Beadle, Nobel laureate in physiology and medicine at Caltech, professor of biology and chairman of the Division of Biology, 1946, 1961. So what I would like to tell you about is how does 1 gene code for 1 enzyme? Now, the key is, of course, the genetic code. The genetic code has the following thing: 3 nucleotides code for 1 amino acid. Why 3? Well, if you had only 1 nucleotide, you could code for 1 amino acid. If you had 2, you could code for 14. If you have 3, you can code for 64. And in 1953, we all know that Watson Crick discovered DNA, the double-stranded DNA. But it took from 1953 to 1966. This is the first genetic code written down fully, except for 1 missing opal up in the upper right-hand corner. And there are 3 nucleotides now coding for 1 amino acid. And the 3 nucleotides are called a codon. And therefore, in the messenger RNA, it's broken up into 3 codons. And that is why the messenger RNA has to be even longer than the amino acid that it's coding for. Now, when the code was written down in 1966 by Crick, he basically said in the first letter of the codon is UCAG, in the second letter of the codon is UCAG, and in the third letter of the codon is UCAG. And you could see here that if you look at GG, it codes for glycine. If you look at GC, it codes for alanine. If you look for CC, it codes for proline. And if you look for CG, it codes for arginine. Now, this is the more recent way of writing it because it has to do with the fact that in the third codon, two of the things, U and C, code for one amino acid, and A and G code for another amino acid, called the wobble. So that's why, in some sense, it's not simply 1, 2, and 3. It's 1, 2, and then it breaks up into the wobble. Okay, that's the code. And so everybody go at Google and load down the genetic code table, and this is what you're going to get. Well, in 1971, I had a headache, and an evolutionary scheme came into my head, in which the basis entered the genetic code in a definite temporal sequence, and correlated amino acids are assigned definite functions in the evolving scheme. The scheme requires a singlet code evolving into a doublet code, GC, coding for glycine, alanine, arginine, proline. The doublet code evolves into a triplet code. Polymerization of nucleotides is thought to have been by block polymerization of triplets rather than a template simple, one nucleotide at a time. The proteins formed were simple structural peptides. No direct nucleotide amino acid stereochemical interaction was required. Rather, an adapter type indirect mechanism is thought to have been functioning since the beginning. So I rewrote the code, GCAU, GCAU, GCAU. That's all I did. Nothing changed. GG coded for glycine, GC coded for alanine, CG coded for arginine, and CC coded for proline. There was no change in the codons and their respective amino acids. So this is written down in a more modern way because I had to change the third codon into the wobble, which is that GA codes for one protein, CU can code for another protein. This is the wobble. So that was the only thing I did differently. GCAU, GCAU, and it's GA, CU, and that's it. Now, looking in that table, I immediately saw something that was very important. That in that table was the evolution of the genetic code. And this is something that is very, very important when you're dealing with historical evolutionary biology. Life has developed its processes, this is St. Georgie, gradually, never rejecting what it has built, but building over what has already taken place. As a result, the cell resembles the site of an archaeological excavation with the successive strata on top of one another. The oldest one, the deepest. The older a process, the more basic a role it plays, and the stronger it will be anchored. The newest processes being dispensed with most easily. In considering the evolution of a complex system, it's useful to adopt an idea expressed by Bernal. In general, the pattern is one of stages of increasing inner complexity, following one another in order of time, each one including in itself structures and processes evolved at the lower level. This I call the onion heuristic. Now, the evolution of the genetic code suddenly appeared from that rewriting of the genetic code. And it starts off with a GC code, going to a GCA code, then to a GCAU code. And here it goes. Here's a GC code, and it codes for the following amino acids: glycine, proline, alanine, and arginine. There is an operational code in the arm of the tRNA. Amino acids added to the genetic code when GCA code comes into existence. Glutamic acid, aspartic acid, glutamine, asparagine, threonine, serine, histidine, and lysine. When A was added to the code scheme, then U had to be added to the anticode scheme in the tRNAs. All these amino acids are hydrophilic. So something is happening because now the hydrophilics are separating from the hydrophobics. The amino acids added when U was added to the genetic code system, valine, leucine, isoleucine, methionine, phenylalanine, tyrosine, tryptophan, and cysteine, except for serine. The amino acids added when U was added to the coding scheme and to the messenger RNA implies that A is added to the anti-coding scheme, the tRNA. All the amino acids added are hydrophobic except for serine. What do I do? I'm sorry. Now, from my last lecture, I talked about the citric acid cycle because my last lecture was on metabolism and it had to do with how you made amino acids, etc. And it came from the citric acid cycle that the number of steps in their biosynthesis from the citric acid cycle was a metabolic metric. And the metabolic metric is the number of metabolic steps it takes to synthesize an amino acid from the citric acid cycle. And that's reference to my previous talk. Now, where am I going to find this wonderful, beautiful hypothesis of the evolution of the genetic code? It has to be in the translational apparatus. That is the apparatus that takes a messenger and converts it into a peptide or a protein. And here is the central actor in this process. There is the messenger RNA going in. Here's the large subunit of the ribosome, and there's the small subunit of the ribosome. There are 3 tRNAs that are reading the message. One of these tRNAs is attached to the polypeptide chain. The next one coming in is the one that has the activated amino acid. And then there's the third one, which is now being tossed out. Now, the big event occurs when the tRNA with the activated amino acid on it is the peptide transfer to it. And therefore, the peptide chain grows by 1 amino acid. Meanwhile, the one that was the peptide chain goes into the exit. This thing is now into the peptidyl site, and now the new tRNA has to come in with the amino acid. Okay, so this is the ribosome. This is the large ribosome and the small ribosome. And the little yellow thing is the 5S RNA. And these blue things sticking on the outside are basically proteins. And here is a deeper picture of what I just told you. There's the exit site. There's the peptidyl binding site that you see is attached to the peptide. And there's the new tRNA called the aminoacyl binding site. So there's a P site and there's an A site, and the transfer will be given from the P peptide to the new amino acid, the new tRNA. So there you are, you have it. The small ribosome is down below where the message is being read, and the peptide is being synthesized in the large ribosome. That's going to be interesting. Now, this shows you a picture of the RNA of the ribosome, a secondary structure. It's about 1,400 or 1,500 nucleotides long, single chain. This is the small subunit one, which is about half its size. It's about 1,400. This is about 3,000. This is about 1,400. Now, the thing I want you to concentrate on is on the PTC site. That is the site where the transfer occurs between the activated tRNA amino acid and the peptide interchange. That's the PTC site. That's where a new peptide bond is being formed. Here is a picture from the group in Georgia Tech, Anton Petrov and Williams. They are the best group in the world. I mean, they have done fantastic work on the ribosome. And there's the PTC site. And these colors are telling you where pieces of the ribosome came in at what time. And so the ribosome grew like an onion from the PTC site. Here is the PTC site taken out. You can see that it's there in the large ribosome. And the greens, I think, form the A site, and the blues form the P site. These are the sites where the so-called ribosome takes place in the ribosome. Now, recently, in 2022, the origin of life was solved because they made a proto-ribosome forms, peptide bonds and links, RNA and protein dominated worlds. Once you cut the separation between the RNA world and protein worlds, you've solved the problem of the origin of life. This, of course, is the RNA world hypothesis. Now, what they did do is that they synthesized this A and P site by themselves, and they then added a system in order to examine our proto-ribosome hypothesis that these two RNAs self-formed and came together. Empirically, RNA chains derived from the conserved pocket region were designed, prepared, tested for their ability to dimerize and catalyze the formation of a peptide bond. Gratifyingly, several of the proto-ribosome constructs led to the formation of a peptide bond between these substrates. To the best of our knowledge, this is the only case in which proto-ribosome hypothesis, which is the one that this is where it started, is directly supported through the activity of a PTC RNA construct. Okay, here's something that is very interesting. It's the history of the ribosome and the origin of translation. Anton Petrov et al., and it's a great paper. Now you'll see here this sort of site of a proto-ribosome occurring. This is something different. This is something that's occurring in a small, for the small ribosome RNA. This thing now evolves into something more complicated, and it begins to make peptides. And I'll tell you about that later. This thing is somehow interacting with something like a messenger RNA. And at some point, you have two protein synthesizing systems, one in the large subunit and another one in the small subunit. I'm not going to talk about the small subunit. I'm going to emphasize the large because that's what we know the most about right now. Then in phase 3, they two come together. This is the small subunit where the message is being read. This is the large subunit where the peptide bond is forming. And now they come together and they evolve into this magnificent thing called the ribosome. Now, the model explains the evolution of the large ribosomal subunit, the small ribosomal subunit, the tRNA, messenger RNA, prokaryotic ribosomes evolve in 6 phases, sequentially acquiring capabilities for RNA folding, catalysis, subunit association, correlated evolution, decoding, energy-driven translocation, surface proteinization. In this model, ribosome proteinization was a driving force for the broad adoption of proteins and other biological processes. The exit tunnel was clearly a central theme of all phases of ribosomal evolution, was continuously extended and rigidified. So we take their word for it, and here's what the 6 phases which they say. Ancestral RNA forms stem loops and many helices. In phase 2, the LSU catalyzed condensation of specific oligomers. The SSU may have a single-stranded RNA binding function, that's messenger RNA. In phase 3, the subunits begin to associate, mediated by the expansion of tRNA from a mini helix to the modern L shape. LSU and SSU evolution is independent and uncorrelated during phase 1 and 3. In phase 4, the evolution of the subunits is correlated. Now, the origin of and evolution of the peptidyl-transferase sites came from proto-tRNAs. So we've gone down the thing right down to the tRNA, and they this is a paper, came out in 2014. We tested the hypothesis Tamura that the molecules of tRNA gave origin to the ribosome, particularly to the peptidyl-transferase center of the 23S RNA. We reconstructed the ancestral sequences from all types of tRNA and compared them in their sequences with the current PTC of 23S ribosomal RNA from different organisms. We built an ancestral sequence of proto-tRNAs that showed a remarkable overall identity of 50.53% with the catalytic site of PTC. We conclude that the PTC was indeed originated by the fusion of ancestral proto-tRNA. And let me show you tRNA. This is the second tRNA that I pointed out to you. It's 70mer, and it's basically built up of 3 subunits. One is the discriminator base, that's D at 73, and then there's a CCA, and that is going to be very, very important. Then there's the acceptor arm, and then there's the T arm, and the T arm is an exact copy of the anticodon stem loop. The D arm is somewhat related, but slightly different. And so those are what the tRNA, it's a small molecule. At the CCA end is where an amino acid is added in an activated form. So this is, again, just to show you that where the discriminator base is. And now, at the end of my talk today, I'm going to talk about the operational code in the acceptor stem. Okay, I'm going to switch now. We've talked about RNA. Let's talk to the ribosomal proteins. These are the ribosomal proteins. There are 78 of them in the eukaryote. There are 68 of them in the archaea. There are 57 of them in bacteria. And there are 34 of them that are universal. That universal means you can take these proteins and you can align them, as you saw what happened in lactase. that's one of the key facts of how you do bioinformatics. So these are the, how shall I say it, the universal ones. And they are found, some of them, at the PTC site. So this is now trying to go from the PTC site and evolve out from the PTC site now, not in terms of RNA, but in terms of the proteins that are associated with the ribosome at these various locations. So they have sectioned the LSU, the large subunit, into concentric shells like an onion, using the site of peptidyl transfer as the origin. This spherical approximation, combined with shell by shell comparison, captures significant information along the evolutionary timeline, revealing, for example, that sequence and conformational similarity of the 23S are greatest near the PT site and diverge smoothly with distance from it. The conformation of ribosomal protein components near the PT origin suggests that they may be molecular fossils of the peptide ancestors of ribosomal proteins. Their abbreviated length may have prescribed formation of a secondary structure, which is indeed absent from the region nearest of the LSU nearest to the PT site. Now, this is what we did. We looked at the proteins and their interactions with the PTC site. The ones that we were interested in is L2, L3, and L4. L22 is now found in the tunnel. L10E is sort of mimicking a third tRNA, actually. Anyways, what we did, of course, was to sequence these universal proteins. And I'll tell you, L2, the things about L2 and L3 and L4, which you will see in which you saw, were of two kinds, and we can go back and see it. There's the globular forms and these extensions. They are totally different. They have come, so it's the extensions that are the fossils. The globular forms are not. There are no alpha helices in the peptide extensions of L2, L3, L4, and L22. The peptide exit tunnel is dominated by the extension of the protein L22, which is a beta hairpin. That's very important. The LSU proteins L23 and 29 are on the surface of the LSU, and these show an increase in complexity in their structure. For example, in L23, aside from a beta sheet, there's appearance of alpha helices. That indicates an increase in the complexity of polypeptide folding. Also seen in L29 is a pure alpha helical protein. L23 interacts with the LSU RNA through a small loop and its globular domain. One sees a progression from the unstructured, positively charged peptides with beta turn loops and beta hairpins to beta sheets and finally to alpha helices. The addition of alpha helices completes the set of primary protein secondary structural elements. It has been pointed out that beta hairpins are likely precursors to the formation of outer membrane beta barrels and related AB and SH3 folds. Both such precursors and structures are expected early on, provided membrane permeability and RNA binding. The AB and SH3 folds are known for their binding of nucleic acids. There is a fold metric: random coils, beta hairpins, beta barrel, alpha beta hairpins, and alpha bundles. Here's what happened when we sequenced the universal proteins. And up there you can see L2, L3, L4. The black ones are found in the archaea. The stapled ones are found in the bacteria. The white ones are found in common, and they are what allows us to align these things. Now, if you look at L2, L3, and L4, the one thing that comes out that these proteins were not put together in one big piece. They were put together by peptides. And there's the universal peptides, and there are the peptides that are unique to the archaea and are unique to the bacteria. And therefore, when you look at these proteins, you could say quite well that they were made up from peptides. But what is even more surprising, this is at the heart of the PTC site. These proteins are different in the archaea and different in the bacteria, yet the genetic code and the ribosomal function are totally invariant over the whole of molecular biology. So it's interesting. There is a lot of history in the ribosomal proteins. But what is interesting is that the taxonomic of these block structures of the universal LSU ribosomal proteins and their extension structures support the idea that these earliest encoded proteins were likely only relatively short peptides and that their more organized globular regions came later. It should be concluded that the extensions of these LSU proteins are associated with the PTC and are involved in LSU early folding, and that they likely represent the very oldest coded, and I don't believe coded peptides, selected for their ability to stabilize the RNA structure. Their universal nature, their association with the PTC, and its assumed antiquity strongly support that they are representative, as Fox, as a friend of mine, has suggested, the very oldest peptides. That there is a fossil information in the ribosomes has been noted earlier, however, with a focus on the RNA. Now, there was, given this sort of evolution of folds from the PTC site to the rest of the ribosome, was then taken, I only looked at it and we inferred it from the exit site, the tunnel. But this fellow, Professor Lupus and Alva, did the following thing. The ribosome is the main survivor of the primordial RNA world. And you can start retracing the steps led to the folding protein today due to its very slow rate of change. Close to the PTC, which is the oldest part of the ribosome, proteins are extended and largely devoted to secondary structure. Further from the center, the secondary structure increases and super secondary topologies come in common, although the protein is still largely lack a hydrophobic core, and it means it's mostly hydrophilic. At the ribosomal periphery, super secondary structures coalesce around hydrophobic cores, forming folds that resembles those seen in proteins of the cytosol. Collectively, ribosomal proteins are thus a window into the time when proteins were acquiring the ability to fold. And this now seems to track the GC code to the GCA code to the GCAU code, where you go from structural proteins to hydrophilic proteins to hydrophobic proteins. And this is also true for random coils, which are structural basically, beta hairpins, beta barrels, alpha beta hairpins, and alpha bundles. So we now have a new metric. We had the metabolic metric, how to make amino acids from the citric acid cycle. We now have a new metric, is how the structures, the coils and the hairpins evolve into the the structured proteins we know today. And this is, he maps this out using the spherical stuff that I showed you on peeling the onion. And this is just what a repeat. Ribosomal proteins repeat these other words, random coils, etc., etc. Structural proteins, hydrophilic proteins, hydrophobic proteins. This seems to track GC, GCA, and GCAU. A model for short, self-assembled peptides in the very early stages of life. Now, this is a field that is now coming into existence, which says it's recently been demonstrated that peptide molecules as simple as dipeptides can self-assemble into well-ordered tubular, fibrillar, and close-cage structures. Other studies have confirmed the ability of dipeptides to act as catalysts and the capability of other peptides, as short as tripeptides, to serve as a template for nucleotide binding and orientation. Unlike complex RNA molecules, the spontaneous formation of functional short peptides in the primordial Earth conditions is very likely. We suggest a novel mechanism for the origin of life that is based on the ability of short peptides to form encapsulated structures, catalyst chemical reactions, and serve as highly ordered template for the assembly of nucleotides. This model may explain the early events that led to the formation of the current biochemical machinery that combines the elaborate and coordinated interaction between nucleic acids and proteins to allow the function of living things. So, what? So I did an experiment with Steve Santos and Shugan Zhang at MIT. We took the self-assembly of peptides of short glycine tails to form nanotubes and nanovesicles. And this is what, this is what we got. And we showed that they self-assembly of these things occur, and it's peptide chains of glycines that are only about 4 to 10 glycines long, and there's aspartic acid put on the end of it, and they form very nice peptide membranes. And this field is exploding. So I'm just telling you that what I saw in the ribosomal proteins evolution is now big time in the origin of life. Prebiotic peptides, molecular hubs in the origin of life. And then this is a very important paper, since they refer to our paper, self-assembly of surfactant-like peptides with variable glycine tails to form nanotubes and nanovesicles. So this is now, life has become an experimental science. Okay, tRNA. Now tRNA, I was told you about the CCA. There's a specific enzyme that adds CCA to the tRNA. It's not encoded in the DNA. And here's what it looks like. This is a head region, and then there's a neck region, and it's done by peptides. The peptides are what is giving you the CCA. It's being read by peptides, and the lower one that has helix G and helix F shows you how the peptides recognize the CTP and the ATP and how they modulate this thing. Okay, now Alan Wiener redid it And he showed that you could make UG or DCC, that you can now start making a whole bunch of different trimers, etc. So I want to get back to the amino acid synthetase. I'm going to go very rapidly on here. This is the enzyme that does the phosphoester formation. This is now a secondary structure of the aminoacyl-tRNA synthetases. They are very, very similar, and they're made out of beta hairpins plus extensions. And this is now the difference between, there's two classes of aminoacyl-tRNA synthetases based on the catalytic site. And the catalytic site is the one that I just showed you with the class 2. This is the one in class 1, and it's based on alpha beta hairpins rather than just beta hairpins, which, as we know. And this is sort of a nice picture showing it of the class. So here is the aminoacyl-tRNA synthetases. And there's the catalytic domain, and there's the anticodon domain. And then there is these editing domains. I'm going to talk mostly about the catalytic domain and the anticodon domain. Well, I told you about tRNA, I told you about the discriminator base, and now we're going to start looking at the anticodon system. Here is how the tRNA interacts with the aminoacyl-tRNA synthetase. The arm interacts with the catalytic site and the editing site when it's there. The anticodon loop interacts with the anticodon module. And this is to show you these two catalytic domains that distinguish them. This is the evolution of the genetic code that I told you about. The GC code proceeding to the extant one is traced out in the evolution of the LSU I showed you, in particular those associated with the peptidyl-transferase site and their evolution to full complex proteins. Now, each one of these aminoacyl-tRNA synthetases codes for 10 amino acids, and the class 1 codes for mainly hydrophobics, but there is, they break up into subgroups. So class 1A is mainly hydrophobics. Class 1B is mainly hydrophilics, and class 1C is hydrophobics. Class 2A is got interesting mixture. Glycine, alanine seem to be first, and then glycine, alanine, and proline come in, and then it's proline, threonine, and serine, and histidine comes in later. And the last one, as 2B is also hydrophilic, and 2C is phenylalanine. So in the GC code, it codes for glycine, alanine, proline, and arginine, DAP, but that's another story. GCA code, aspartate, asparagine, lysine, glutamate, glutamine, and lysine, threonine, serine, histidine. It's hydrophilic. And GCAU code is valine, leucine, isoleucine, methionine, phenylalanine, tyrosine, tryptophan, and cysteine. And here's the evolution of the genetic code related to the metabolic metric. And there were 3 different areas of the metabolic metric. And the GC code is related to group 1 of the metabolic metric, and GCA was group 2 of the metabolic metric, and the GCAU was related to the group 3 of the metabolic metric. Now, there's a very important paper that I wanted to tell you about. It's called The Amino Acid Evolution of the Genetic Code and Aminoacyl-tRNA Synthetases. And there you could see, right there, the relationship of the GC code to the GCA code and to the GCAU code. And it all has to do that how many steps from the citric acid cycle is involved. So glycine alanine is only one step because it's pyruvate and glyoxylate. Proline is 3 steps from glutamate. Arginine is 8 steps. So arginine is a late comer. These are now class 2, are aspartate, asparagine, and all of these are all in the second GAC group. The only ones in the GAC group are glutamate and glutamine, and one of the lysines. And then, as you see in the GCAU group, the new amino acids coming in are all in class 1 of the synthetases. So there's a relationship between the GC code, GCA code, and GCAU code with respect to the aminoacyl-tRNA synthetases. Based on tRNAs, it has been proposed that the earliest codons were associated with GC triplets, and the 3 evolutionary stages established the modern form of the genetic code. The earliest stage is assumed to be from GC bases only, whereas the later stage of evolution into 3rd base was added to GCA. Finally, U was added, establishing a 4-letter code, Hartman, 1995. It is very likely the records of these stages are retained in the evolutionary history of the aaRS. This is coming from that paper. Okay, in Schimmel, now I have to talk real fast. In Schimmel, in 1993, he found an operational RNA code for amino acids and possible relationship to the genetic code. And the question was, this is holy moly, because the code is really lying in the anticode, and that is lying in the stem loop of the anticodon system. And here they find a code in the arm of the thing that is recognized by the synthetases. So they then tried all kinds of things, and they were all being recognized by this aminoacyl-tRNA synthetases. And this code was in the arm. Now, the people that then went and looked at it much more carefully was Roda and Ono. And in their paper, they showed that in distinction to the single-stranded anticodons built of GCAU bases, they're presumably double-stranded precursors at the first 3 positions of the acceptor stem are composed almost invariably of GC and CG pairs. Thus, the second operational RNA code responsible for correct amino acylation seems to be a GC code preceding the classic genetic code. Although historically rooted, the 2 codes were destined to and they then say the archaic GC code was hypothesized a long time ago. The general thesis has been argued that, like all other evolving systems, the system began simply and evolved to the present complexity. The very fact that the anticodon-like triplets in the acceptor helix turn out to be composed of predominantly GC and CG code strongly supports this hypothesis. So there came a huge thing that said that the GC code was involved in the earliest stages of the evolution of the genetic code. Now, Roda and Ono then set out to show how this could occur, and they had building triplets using basically ACCA and UGGU. And then they had coding triplets. And the other ones were, here, this is DCCA and DGGU. The substitute for the D's, U in the small d and A in the large d. And they then began to build up their tRNA from these things. And they built up the arm, and then they built up a loop. And they therefore showed how this thing would essentially be related to what they were finding in the arm and the loop, in the arm and the GC code in the arm. So that takes us down to where this is the end of the thing, because the next chapter would begin with the smallest subunit of the ribosome. So I have a myth. And my myth is that life began with Father Sun and Mother Earth, and which gave birth to self-reproducing iron-rich clays who evolved up metabolism, including a Thioester world. And the Thioester world evolved up the genetic code when phosphate entered. And that has to do with the code evolving in the transition from the Thioester world to a phosphoester world. So many times people ask me, well, isn't this related to the Bible, starting with clays? And I said, well, it's slightly different. My hypothesis is that iron-rich clays started up and evolved up man, and man evolved up God. So clays evolved up man, and man evolved up God. And that is my usual answer to saying that that in the Genesis, God made man out of clay. That's it.
Okay. Okay, thank you. Any questions? Any questions from the audience?
I say,
All understood.
it's all understood, it's simple.
That was a great talk. Really enjoyed that. Thanks.
Anyways, this is an experiment of trying to give a molecular biology talk to an audience that I assume does not think too much about molecular biology. But the ideas are very, very simple. They're just trying to unpeel the onion. And the onion is still there. It's very much like geology. It's very much like an archaeological dig. And so all this is, all I have done is, according to St. Georgie, is an archaeological dig. And that's what I was trying to tell you about.
Okay, this sounds great. Thank you. Let me save the video. Okay, so that you can have it for your record.